Good morning, everyone. Welcome to the second day of the EPTRI Open Meeting. We have a full day in front of us, and there will be a lot of science and interesting presentation. I hope we will enjoy the day, but it's a long day. We will have quite a lot of audience still signing, so I'm trying to keep the timing, but to allow also other people to join. So give them a little bit of time. As soon as I see that the number is not increasing anymore, we will start.
Okay, we have almost 80 participants connected, so I think that it is.
Yeah, it seems that the number is stabilized. So we started the first session. We have a session dedicated to scientific presentation to see how many different kind of contribution can be added inside the future infrastructure. So it's my pleasure to start with Professor Diaz-Manera on human fibroadipogenic precursor cells, and let's see how he wants to use them. Professor Manera, it's your time.
Hello, can you hear me? Yes?
Yes.
So? Yeah, okay. So good morning to everyone. I would like to thank the organizing committee for allowing me to present my data. My name is Jordi Diaz-Manera, and I am an adult neurologist working at the John Walton Muscular Dystrophy Research Center in Newcastle. And I'm going to talk to you about the human fibroadipogenic precursor cells as a new tool for investigating antifibrotic drugs in muscular dystrophies. This is a collaborative work that was mainly done at my former institution, that was Hospital Sant Pau in Barcelona. So next slide, please. Can you go to the next slide? Yes, okay. So as you may know, muscular dystrophies are rare hereditary disorders produced by mutation in genes that codify proteins of the muscle fibers. There are 32 different genes described so far, and in general, all patients develop muscle weakness that progresses over the years, leading to a variable degree of disability, from patients that need a cane for walking to patients that are completely wheelchair bound and need help from their relatives to do most of their daily life activities. In the slide, you can see an example of one of the most dramatic diseases, which is Duchenne muscular dystrophy, which onset is at 4 or 5 years of age with difficulties running. Then the kids progress, and at 9, 10 years old, they have a clear muscle weakness involving proximal muscles of the lower limbs with problems to climb upstairs. Early in the adolescence, they need orthosis to walk, and as this guy that you can see here in the photo that is wearing calipers, and they lose ambulation before 20 years old. From that moment, there is a progressive involvement of the heart and the respiratory muscles, which are the main cause of death in these patients. So next slide. From a histological point of view, muscular dystrophies are characterized by a progressive loss of muscle fibers. So the absence of proteins leads to, in most of the cases, instability of the muscle membrane, which is damaged after every muscle contraction, producing muscle inflammation and necrosis. The loss of muscle fibers is associated to a progressive increase in the amount of connective and fat tissue, which in the end replaces the whole muscle and has not contractile properties, being therefore the main cause of muscle weakness. In the last year, fibroadipogenic precursor cells, also known as FABs, have been described as the main protagonist in the process of fibrofatty expansion. Next slide, please. So FABs are muscle-resident mesenchymal stem cells able to differentiate to profibrotic or proadipogenic cells. After an acute muscle damage, they activate, proliferate, and release components of the extracellular matrix, contributing to the regeneration of muscle. So it seems that these cells are important for muscle homeostasis. Recent investigations have shown that after the muscle is completely regenerated, they enter in apoptosis and die. But during chronic muscular damage, FABs are continuously activated, leading to fibrosis and fat deposition in skeletal muscles. So we have observed that the number of FABs increases progressively in muscular dystrophy muscles, correlating with the amount of connective tissue present in the muscle, suggesting that they have a predominant role in this process. So next slide. Our hypothesis with this work was that human Fab cells could be a good model to test drugs counteracting its pro-fibrotic and pro-adipogenic potential, and we had 4 different objectives. The first one was to isolate human Fabs from frozen muscle samples. The second one was to compare the differentiation ability of these cells with freshly isolated Fabs. Then we also aim to immortalize isolated Fabs for further use in experiments, and finally to test the ability to decrease Fabs pro-fibrotic activity using antifibrotic drugs. So next slide. So far, the protocols published recommend isolate Fabs from fresh muscles. Most of the papers published uses Fabs from mice that are obtained after sacrificing the mice. In humans, Fabs are isolated from fresh muscles of patients that are being operated, for example, in the traumatology department, and are donors. In the case of human, fresh muscle is digested and plated in cell culture plates, expanded, and Fabs are obtained using sorting for the PDGF receptor alpha, which is the marker typical of these cells. This process limits the access to human Fabs because you need fresh mass. So in our biobank in Barcelona, when we receive a muscle biopsy for diagnosis, we split it in 2 parts when possible. One part is for diagnosis, and the other is minced in small pieces and stored embedded in growth medium in liquid nitrogen. This allows us to play these pieces even many years after obtaining the biopsies and grow cells from it. Therefore, we took advantage of this process to isolate Fabs. We plated explants from controls and from muscle biopsies used for diagnose patients with different muscular dystrophies, including Duchenne muscular dystrophies. We grew cells, obtained a mixed population, and sorted PDGF receptor alpha cells, which are Fabs. As you can see, there were much more Fabs in the Duchenne than in the controls. I am showing here the cytometry panels. And this is in accordance with our previous investigation, showing that Fabs are increased in Duchenne. This approach allowed to isolate cells from patients in which we already know the diagnosis, because the diagnostic pathway has been done, and therefore we could study the specific properties of whatever disease we want, as long as we store explant in these conditions. After isolating the cells, we passed them several times and analyzed the presence of PDGF receptor alpha, which define Fabs, to be sure that they continue expressing this receptor. The receptor was present at low, from 1 to 4 passages, medium, from 5 to 8 passages, and a high number of passages, more than 10, meaning that these cells maintained their more essential feature, the expression of this marker for long periods of time. Next slide. We have also developed a protocol to differentiate human Fabs to adipogenic and fibrotic cells. Using a cell medium known as a STEMPRO, you induce differentiation to Fab. As you can see, cells started to accumulate large amount of fat in vesicles after 9 days in culture, as demonstrated by oil red staining, but also using other staining as lipitox or perilipin staining. To differentiate the cells to fibrotic line, we add PDGF-AA to the medium and observe an increase in the amount of collagen in the wells. So these 2 different approaches will allow us to test different anti-adipogenic and anti-fibrotic drugs in these human cells models. Next slide. However, I would like to remember that these are primary cell lines, and as you know, these cells enter in senescence after a number of passages. In the case of Fabs, we observe this after 12 passages, losing their ability to proliferate or differentiate. Therefore, in collaboration with Dr. Mouli, who works at the Institut d'Immunologie in Paris, we decided to immortalize the cell, transfecting 2 different genes, the cyclin-dependent kinase 4 and the telomerase reverse transcriptase gene, using adenovirus. After that, our immortalized human Fabs were able to proliferate continuously without losing the differentiation abilities. This process is important because it allows us to use these cells as many times as we want without losing them with the time. Next slide. This is just an example of what we can do with these cells. We are here testing a new drug in order to slow the degenerative process of the mast cells. Here, what we are doing is to test a drug that we are going to call drug X. Panel A show proliferation assays with and without drug X. As you can see, the drug reduced proliferation of FABs induced by PDGF-AA at 48, but especially at 72 hours. Panel B showed that drug X reduced migration of FABs induced by PDGF-A at 48 and 72 hours. Panel C showed how the drug reduced the release of collagen by FABs induced by PDGF-A. And finally, panel D showed how the drug X reduced expression of actin, which is a cytoskeletal protein commonly expressed by FABs when they differentiate to fibrotic cells. In our opinion, these cells could be used to test large batteries of antifibrotic and anti-adipogenic drugs in vitro in order to know if they could be good candidates for being tested in animal models. So next slide. So as a conclusion, we have been able to develop a protocol to isolate FABs from already frozen muscle biopsies of patients with muscular dystrophies and control. These FABs can be immortalized easily and effectively, and therefore can be used in further experiments. And human immortalized FABs can be used to test the effectivity of antifibrotic and anti-adipogenic drugs in vitro, analyzing their collagen-1 expression and the production of fat. I would like to thank all the people from my former lab in Barcelona, especially Xavier Suarez and Esther Fernandez, who have been working in this project. Thank you very much for your attention.
Thank you very much for this nice presentation. Is there any questions? To do questions, you have a box in the GoToMeeting application, so you can write it there. So no question for now. Just a question then from me, just to start and take away the afraid people from the time. You have a nice system based on cells. However, is this applicable for a high throughput screening of existing drugs? So can you adapt it and use in a automated system?
Yes, of course. The good thing about this is that you can, for example, use it. This is what we are doing now. We are now testing a battery of already known drugs that can be antifibrotic. And what we have done is that we have plated the cells in a multi-well, 96 multi-well plate. So we can test even 96 drugs at the meantime. And what we are just analyzing, for example, in this approach is the amount of collagen that cells express with and without the drug. So it's a good system because it allows you to test how many drugs you want in a very easy manner. Obviously, these are cells. So this means that you can, for example, identify some drug that can be interesting because they are decreasing the collagen expression. But after that, you can do also more experiments to test proliferation, migration, or even move into an animal model with this drug.
Well, I see a lot of possible application here, so it will be interesting to follow your system and see how it evolves with time. You know, my point of view is always to find a way to pay for it, but that's another way to see things. Thanks a lot, Manera. And we will see now the next speaker, Professor Verpelli, talking about the characterization of two genes in Phelan-McDermid syndrome. Professor Verpelli, it's your time.
Okay, I want to start saying thank you to the organizer for allowing me to present the data obtained in our lab. So, Phelan-McDermid syndrome, next. Okay, next, please. No, before, okay. Phelan-McDermid syndrome is a neurodevelopmental disorder characterized by global developmental delay, absent or delayed language, ASD behavior, a strong form of neonatal hypotonia, normal to advanced growth, and more than a quarter of patients show a severe form of epilepsy. The syndrome is caused by a deletion on chromosome 22q14, and the size of the deletion is extremely heterogeneous among patients. So different patients show different gene deleted in them. Next, please. So among these genes, SHAN3 gene is considered one of the major causes of the neurological symptoms found in patients with PMS. SHAN3 encodes for a huge scaffold protein composed by several protein-protein interaction domains that allow SHAN3 to interact with more than 30 synaptic proteins. So in our lab, we want to study the role of SHAN3 in neuronal development and brain function. To do this, we generated, in collaboration with Tobias Boker in Germany, a SHAN3 knockout mice. We start analyzing the behavior of these mice. I can probably next. Okay. We start analyzing the behavior of these mice, and we found that we found that the mice present repetitive behavior, reduced sociability, and impairment in spatial learning and memory. So this means that these mice represent a good model to study the behavioral symptoms found in the kids that have PMS syndrome. Next. Through its proline-rich domain, SHAN3 is able to interact with another scaffold protein, Homer, and the formation of the complex SHAN3 Homer bind type 1 glutamate metabotropic receptor with its intracellular signaling pathway, leading to an increase in calcium release in response to glutamate activation. So we start analyzing the interaction between Homer and GLU5 in absence of SHAN3 by doing immunoprecipitation in synaptosomal preparation. And we found that specifically in striatum and cortex of knockout mice, there were a reduction in interaction between Homer and GLU5. So we want to analyze the activity of the receptor in these two brain areas. And when we stimulated cortical neurons with DHPG that is able to specifically activate GLU5, we found in cortical neurons derived from SHAN3 knockout mice a significant decrease in intracellular calcium release. The other brain areas that seems to be affected by SHAN3 absence is striatum. So we analyze the electrophysiological properties of striatal neurons. And while in wild-type neurons, co-treatment with NMDA plus DHPG cause a significant increase in NMDA-mediated currents, in SHAN3 knockout neurons, we were not able to see this increase in NMDA-mediated current. So this data suggests that the SHAN3 absence cause a significant decrease in GLU5 expression and function in striatum and cortex. Importantly, when we treated mice with a drug able to activate GLU5 receptor, we were able to rescue this defect. So the idea was to use a drug able to activate GLU5 to rescue the behavioral defect that we found in mice. So we use a new drug that is more safe and without side effect compared with the other GLU5 PAM. And we acute treated SHAN3 knockout mice, sorry, before. Previous, okay. And we found that acute treatment with this new PAM was able to rescue in a dose-dependent manner all the behavioral defects that we found in mice. So we rescue repetitive behavior, sociability, and cognitive rigidity that are the three core symptoms of ASD. Next. So in conclusion, our data show that SHAN3 deletion impair Anglo5 synaptic localization and function, that the ASD-like symptoms that we found in SHAN3 knockout mice that seems related with this Anglo5 dysfunction can be rescued by treatment with Anglo5 PAM. And importantly, taken together, these data suggest that Anglo5 may represent a good candidate for studying new pharmacological treatment, and that treatment with Anglo5 PAM may represent a future therapy for kids affected by PMS and SHAN3 deletion. However, I told you that SHAN3 is not the only genes deleted in Phelan-McDermid syndrome. So we want to study also other genes. And among them, Sultan4A1 is deleted in about 30% of patients with PMS. Why we choose Sultan4A1? Because Sultan4A1 is selectively expressed in neurons. Polymorphism in Sultan4A1 were linked to the increased risk of schizophrenia, and decreased level of Sultan4A1 were found in patients with bipolar disorder. We start analyzing the level of Sultan4A1 during development in vitro and in vivo, and in both cases, we found an increase of the protein expression during development. In particular, in vivo, we found that Sultan4A1 reached a high level around postnatal day 30. And importantly, this level remain high also in adulthood, suggesting that Sultan4A1 may have a role, an extremely important role during neuronal maturation, but also a role in adulthood. We also tested the level of this protein in different brain areas, and we found that Sultan4A1 is highly expressed in hippocampus, striatum, cortex, and cerebellum. Next. Next. To clarify, to study the role of Sultan4A1 in neurons, we designed a specific shRNA for Sultan4A1, and we found that knockdown of this gene in neurons cause a reduced neuronal arborization and cause a significant decrease in spine density. So we want to analyze what happened at the molecular level. And to do this, we analyze the expression of a panel of synaptic protein in synaptosomal preparation. And we found in neurons knockdown for Sultan4A1 a significant decrease in NMDA receptor expression. These data were also confirmed by immunofluorescence, in which again we found a significant reduction in positive PANTA for NMDA receptor in neuron knockdown for Sultan4A1. So our idea was that Sultan4A1 may modulate the activity of NMDA receptor. And this hypothesis was confirmed by electrophysiological recording, in which we found a significant decrease in NMDA-mediated current in Sultan4A1 knockdown neurons compared with wild-type neurons. So a protein that is linked to Sultan4A1 that bind Sultan4A1 is PIN1. PIN1 is another scaffold protein expressed at synapses, where it exerts a negative effect on NMDA receptor activity by blocking the formation of the complex NMDA receptor PSD95. So we analyzed the expression of PIN1 in our neurons knockout for Sultan4A1, and we found a significant increase of the expression of PIN1. So our idea is that the defect found in abscess of Sultan4A1 may be due to an increase of PIN1 expression and activity. Next. Next. So to test our hypothesis, we treated a neuron with a specific PIN1 inhibitor, that is a PIB, and we found that the treatment with PIB were able to completely rescue both NMDA current in sultan-4 knockdown neurons, and also were able to restore to normal level the number of dendritic spines. So our hypothesis, in conclusion, is that sultan-4 A1 is able to modulate NMDA receptor activity by binding PIN1. So sequestering PIN1, sultan-4 A1 facilitate the formation of the complex NMDA receptor PSD95 that is essential to the function of the receptor. At the contrary, in absence of sultan-4 A1, PIN1 is able to interact with the PSD95, so reducing the probability of the formation of the complex NMDA receptor PSD95, so reducing the activity of the complex. So taken together, this data suggests that also sultan-4 A1 may be related to the symptoms found in kids with Phelan-McDermid symptoms, and also that PIN1 may be another pharmacological target to develop the new treatment for a patient affected by Phelan-McDermid. I want to thank you, all the people that work with me in this project, and you for your attention.
Thank you, Professor Verpelli. I had to find again in my boxes that was hidden somewhere. Any question? I don't see any question now in the box in the system, but I will make my comments anyway. Professor Verpelli, did you observe any alteration in behavior in these mice?
In which, sorry, in the
Behavior.
we have only the SHAM3 knockout mice, and in these mice we found a strong alteration on behavior. They show all the major symptoms of ASD. Unfortunately, at the moment, we do not have the knockout mice for sultan-4 A1, but we would like to generate also them, and we expect to find some behavioral defect. In particular, some sultan-4 A1 knockout mice were generated in a lab in USA, and these mice, the knockout mice, die around postnatal 1, postnatal 7, so suggesting that the sultan-4 A1 also is extremely important during neurodevelopmental and also is important for the formation of neurons.
Okay, we will see with the interest your future results. Thank you very much.
Thank you.
Professor Van Kruchten, and in mini pig model, the Göttingen mini pig. Let's see how this can be used for drug development into pediatric population.
Thank you, Giovanni, for the introduction. So as you heard yesterday afternoon in the presentations of the thematic research platforms, there is a clear need for pediatric animal models, especially for drug discovery, but also for developmental pharmacology. So in this short presentation, I would like to highlight some of our findings with the neonatal and juvenile Göttingen mini pig in view of pediatric drug development. So next slide, please. Dose precision in pediatric patients up to 2 years of age is challenging, and in this respect, pediatric animal models can be very valuable, not only from a safety point of view when developing new drugs, but also to explore disease conditions in the pediatric population. And as such, potentially we can find new drug targets, as you heard yesterday. In our research infrastructure, we are using the neonatal and juvenile Göttingen mini pig as an animal model, and this for 3 different purposes, which is in the next slide. So neonatal and juvenile gutting mini pigs are used in first instance to better predict exposure of small molecule drugs in the pediatric population, and this to avoid over or underdosing of patients. And therefore, we gathered several physiological data in the neonatal and juvenile gutting mini pig in order to develop a so-called physiological-based pharmacokinetic model, so a PBPK model. This model can then be made available to several industry and academic groups for better dose setting in preclinical and also clinical studies. As a second bullet point, you can see single-stranded oligonucleotides. So besides small molecule drugs, we are also investigating these SSOs in neonatal and juvenile gutting mini pigs. And these SSOs are new drug modalities. They are intended to inhibit the translation of proteins, for example, in cancer. And typically non-human primates are used as the non-rodent model of choice. However, when we have a look at these new drugs, we need to take into account that also the gutting mini pig can be a very good model. And this is due to the fact that they can represent also this condition, or they can be a good model for these types of compounds. So this has been shown already for the adult gutting mini pig, and we are currently qualifying the juvenile gutting mini pig for pediatric safety testing of SSOs. And finally, we also investigated disease states as a third bullet point in the neonatal gutting mini pig. And in our facility, we are currently investigating perinatal asphyxia in this model. And more specifically, we are inducing this condition in newborn piglets, and we will assess the pharmacokinetics of several drugs in this condition with and without cooling therapy, as is done in the critical situation. So the ultimate aim is to assess whether cooling and or the disease state affect the exposure of the compounds, and whether dose setting needs to be adjusted in this clinical condition. So if you go to the next slide. To achieve these objectives, we are using gutting mini pigs of several age groups, and on the left side you see the column with the different age groups. So we start immediately after birth, so day 1, and then up to day 28, which is the weaning age. And these age groups actually correspond roughly to an ultimate age of 2 years of age in the human population. Besides these age groups, we have also included 2 fetal stages, so 84 and 86 days of gestation and 108 days of gestation. And this is to cover the premature babies in the human population. And finally, we've also included adult references as a reference value, of course. And as you can see, we also included both genders. So at this point, we have measured several parameters in these age groups, such as the pH and the length of the gastrointestinal system, organ weights, but we also assessed drug metabolism, and this from an activity point of view and also abundance. And we mainly focused on phase 1 and phase 2 enzymes. And all these data can be fed into a PBPK model, as I already mentioned before. So in view of the SSOs, we assessed the ontogeny of exo and endonucleases, as the unmodified SSOs can be actually hydrolyzed by these nucleases. So they are not prone for phase 1 and phase 2 metabolism, but they can be hydrolyzed by endogenous nucleases in the blood and in organs. And in our in vivo study, we also assessed toxicity by several parameters, such as clinical observations, clinical chemistry, body weight, etc. And so finally, we set up an asphyxia model with and without cooling therapy, as I mentioned in the slide before. So in the following slides, I will take you through some of the results. So if we have a look at this slide, this is showing you some of the morphometric data. And what we noticed, as in the human pediatric population, we saw in the juvenile gutting mini pigs a fast decrease in gastric pH from postnatal day 1 up to postnatal day 28. And this, of course, needs to be taken into account when you look at your drug, because this can have an impact on the absorption of your drugs depending on their physical chemical properties. We also noted high relative organ weight during the first week of life, and this was then followed by a gradual decrease up to postnatal day 28. We saw a fast increase in absolute organ weight, which continued up to 3 to 4 months. And there was a linear increase in body weight that was continued up to 5 months, and this was actually expected. If you click, all these data can then be fed into a PBPK model, but this is of course not sufficient. We also need drug metabolism data. These are one of the most critical data in a PBPK model, and this is shown in the next slide. So this is a slide in which we see 4 graphs, and the x-axis of each of these graphs is actually depicting the age groups. As you can see, the ones that I mentioned before, and the HLM stands for human liver microsomes. So we included also a control, and these were the human liver microsomes. In the y-axis, you can see the formation of different metabolites. So on the left upper panel, you see the formation of acetaminophen, which is the CYP1A2 metabolite of phenacetin. On the right upper panel, you see the formation of 4-hydroxytolbutamide, which is the CYP2C9 metabolite of tolbutamide. Left below, it's the formation of dextrofan, so the CYP2D6 metabolite of dextromethorphan. And the last one on the left, on the right bottom, you can see 1-hydroxymidazolam, the CYP3A4 metabolite of midazolam. Now, if you look at all of these graphs, you can see that there is a clear ontogeny profile for the formation of these metabolites. So that's for all of them the case. But if you then click, and click again, then you can see that we have for 2 of them actually a fast onset of drug metabolizing capacity. And if you click again, then you see on the right side that we have a slower onset of drug metabolizing capacity. So it's important to take this into account for the different enzymes, that there may be a different maturation profile. And this, of course, can again have an impact on your drug and potential overdosing of your drug when we are talking about the youngest age groups. We've also assessed this for phase 2. I have not included the data in this graph, but if you click again, then you can find the reference with all the data below. If you go to the next slide. So, as I mentioned in the objective, we are also qualifying the neonatal and juvenile Göttingen mini-pig for safety testing of SSOs. And the adult one has been already done in 2017, as you can see on the left upper panel. And we are currently now qualifying the juvenile Göttingen mini-pig. If you click, please. You can click again. So for the SSOs, we have not found any overtoxicity, not in the young animals and also not in the adult ones. And I've just included one graph just as an example, because I cannot show you all the data and they're also still preliminary. I've just included in this graph actually the data for the ontogeny of the nucleases. And what you can see on this graph is actually 3 nucleases that are included. You can see it in the x-axis below for 3 different age groups. And to keep it simple, also for this graph, you can see that there is an ontogeny profile, and this again needs to be taken into account when you are exposing your young animals, because for some of the nucleases, they still need to undergo a maturation profile, and by this dose that you give, you can actually overdose your animals. So that's actually the key message that I want to give for this slide. If you want to have more data, if you click again, you can find it back in the reference on the left side. So to conclude, I want to show you some data in the next slide on the asphyxia model. So in our group, we are currently developing a neonatal asphyxia model in the Göttingen mini-pig, and this model has already been established in the conventional piglet, as you can see on the left side. As pharmaceutical companies are using the Göttingen mini-pig, we want to establish it in this strain. And what we will do, if you click again, We will modify the setup from our Danish colleagues, and you can find the reference below, in which we will actually assess cooling. So if you click on the pharmacokinetics of different compounds, so if you click again, we will assess the effect of cooling on 4 compounds: midazolam, phenobarbital, topiramate, and fentanyl. And these are 4 compounds that are used in the clinical ICU unit, and they also have a different metabolism profile. So that's why we chose those 4 compounds. So what we will do, if you click again, we will assess the pharmacokinetics, and we will then also feed this back into a PBPK model in order to better predict exposure in this population, and also to have a better dose precision, because that's actually the aim in the end of this research. So if you then go to the next and last slide. So to conclude, so far we have already shown that drug metabolism of small molecule drugs and also gastrointestinal parameters are similar between neonatal and juvenile Göttingen mini pigs in comparison with the pediatric population. Next. Second, the neonatal and juvenile Göttingen mini pig also appears to be a valuable model for new modalities, such as the SSOs. And third, there are also opportunities for assessment of covariates that cannot be addressed in a clinical setting, and I've used the perinatal asphyxia model as an example. So finally, I would like to thank all our, in the next slide, I would like to thank all our academic and our industry partners, and also our Flemish funding agency for their trust in our group and their support. And I would like to thank you for your attention.
Thank you very much, Professor Van Kruchen. So in your opinion, is the juvenile pig model going to be one of the best approach, since we cannot use any more primate models and so on?
I think if we talk about non-rodent models, then indeed the Göttingen mini pig will be one of the mostly used ones.
Is there any strain differences?
When you look at drug metabolism, so we compared actually also our data with the conventional pigs. So this has been done by a group in Ghent. And there are some differences in the ontogeny profile for a certain number of enzymes. But roughly taken, the conventional pig and the Göttingen mini pig are quite similar.
Okay. Okay, thank you very much, Professor Van Kruchen. We have a problem with the next speaker that is not able to connect. In the meantime, we will project the file with the audio, so you will hear his presentation. I remember everyone, there is a box in the GoToWebinar control panel where you can put down your question for the speaker. So let's start the audio from, yes?
There are 3 questions from the speaker, from the audience for the speaker.
I don't see them, so.
Okay, I will read them for you, for the speaker. Is there a pathophysiology of your models similar to those of neonatal?
You are asking to Van Kruchen?
Yeah, of course.
Yeah, sorry, I was on mute. So did I get it correctly that there's a question regarding physiology comparison between the pig and the human?
I will repeat the question for you. Is the pathophysiology of your model similar to those of neonatal?
Okay, so that's referring to the perinatal asphyxia model. So there may be some differences because we are actually inducing it postnatally. So it's not something that we will do in utero. So that's something that we need to take into account. But I think we will be able to extrapolate our findings on pharmacokinetics to the pediatric population.
Thank you.
Okay.
There is an
other question, Valeria?
Yeah. There is an observation from Evelyn Jacques-Gran that said, thank you for this presentation, as you showed the different maturation profile of the key steps of drug disposition. There is another question that is, how this may impact extrapolation to the pediatric patients?
Yeah, so that's, I think, referring to the PBPK model. But I assume that, yeah.
It is referred to the maturation profile of the key steps of drug disposition that are some of them different in humans.
Yeah.
And so the question is, may impact extrapolation to pediatric patients?
Yeah, so what you actually do with this model, so you can actually build it for your preclinical species, and that's also what's done actually for the human population. So there will, of course, be differences. But if you have all your key parameters in the system, you will be able to compare both species. So it will never be a one-to-one extrapolation, that's impossible. But if you have a good solid model also for your preclinical species, and you know the difference with the human population, you can actually adapt for this. So you can include correction factors.
Okay, thank you so much. There is another last question that is, what about using this model assess different in bioavailability in neonates?
Sorry, I didn't get that one. Could you please repeat?
What about using this model assess difference bioavailability in neonates?
I'm not sure if I get the question.
I will try to repeat. What about using this model assess difference bioavailability in neonates?
So you mean that there would be differences in bio?
Go ahead.
So that there would be differences.
I think that we can ask to the requiring person to write again the question with more specification in the meantime, and then in case we will discuss about this question after.
Okay, thank you.
Thank you so much, Steven. Now we can go to the
Thanks,
next
Valeria.
presentation.
Yes, start the presentation, please. Sorry for the delay. We try to solve the audio issues. For giving me the opportunity to present the first steps of a project aimed at implementing in silico methodologies and tools for predicting developmental toxicity in a platform of drug discovery in pediatry. Actually, these tools may have potential applications not only in drug discovery for pediatric drugs, but also in drug formulation science for pediatry, of course. Drug design in pediatrics, discovery and development drugs, medicines for children still represents a real-life open challenge. We need to improve in the platform of drug-specific drug design, age-related drug design, the methods and models for property-based drug design and optimization, models for predicting, models, methods, and tools for predicting and especially metabolism-related toxicology prediction, the methods of computational toxicology. We need to improve these tools, methods, and tools at the early phases of drug discovery to have a chance for designing and producing drugs appropriate for pediatrics. Just to have an idea of the impact of just look at the effect of pH-dependent ionization drug solubility, and drug solubility, and drug solubility is the most important factor related to ADME profile of drugs. Just look at the effect of pH-dependent ionization of drug solubility, for example, and it is known that media, pH, and buffer capacity has a very strong impact on drug ionization and drug solubility, and solubility of compounds and gastrointestinal contents can be affected by other factors, such as fat or micelle formation. Just to have an idea of this, please give a look to the average-dependent factors that influence the in vivo solubility, dissolution of oral dosage forms, and subsequent drug absorption. For example, just the pH of gastric fluid. It changes during the different phases of age from newborn to infant to child and adolescent. And this change may have a strong impact on the absorption of drug, on dissolution of them, solubility, in vivo solubility, and also in preparing dosage forms. Metabolism in pediatrics is important to be taken into account just already at the phases of drug discovery for pediatrics. The activity of a drug metabolizing enzyme or drug metabolizing enzymes is low at birth and reached adult by early childhood. And children in general have a large liver size and hepatic blood flow for body weight as compared to adults. And this tends to increase hepatic clearance of chemicals when enzymatic activity is similar to that observed in adults. So developmental toxicity and the methods for predicting toxicology is important, and our aims is to develop a computational platform for in silico prediction of age-related drug metabolism studies, drug and toxicity, even improving and implementing new data in the existing database and tools. Several steps are predicted in our project. The first one is the overviewing and assessment of in silico methods currently available for predicting developmental toxicity. The second step is the data gathering from recent high-quality database. Some examples are reported in parentheses. For example, QSAR tools release step, which stands for safety and toxicity excipient for pediatric databases. And this is an important database for developing formulations for appropriate formulation for pediatrics, excuse me. Derivation of new developmental toxicity model based on machine learning and artificial intelligence, and finally endpoint correlation studies. Here, a database, one of the most recent developed database for prediction of metabolism, not in general for metabolism of drugs, is shown. This database, MetaQSAR, is proved to be quite reliable in predicting metabolism. Maybe this can be a good basis for implementing data and information, so to improve the prediction of age-related metabolism and metabolism-related toxicology. This MetaQSAR was published in Journal Medicinal Chemistry, but by groups of authors from Milan, Professor Vistoli and Pedretti, and with the overview of Professor Bernard Testa. Here, so the quality of data is ensured by manually created meta-analysis of the selected specialized literature, such as chemical research in toxicology, just some primary sources of data, metabolic data, drug metabolism disposition, xenobiotics, and other. And the second important point of development of this meta-QSAR database is a novel detected classification of the metabolic reactions. And maybe from a molecular point of view, this is a real strength point of the real strength of this. Here some important information about this database are reported in terms of high number of molecules included, reactions, enzyme classification as sources of and selection, of course. And here, just the classification of metabolite reaction, metabolism reaction classification from the molecular point of view. As a matter of fact, the first proof was that several molecules, not metabolism for molecules not included in the database, are predicted quite well on the basis of similarity analysis. As you can see here, some drugs are included, such as cobimetinib or ositinib, which are very recently approved drugs by FDA and the European Medicine Agency for respectively for metastatic melanoma with specific mutation or non-small cell lung carcinomas with specific mutation. Fimacetan, for example, which is approved for hypertension and heart failure, which was approved, drugs is a sartan approved for hypertension and heart failure. And, for example, atomoxetin, which is approved treatment of attention deficit and hyperactivity disorders, and other drugs, for example, for pain, for treatment of pain, or for treatment of chronic myeloid leukemia. So the results are quite good. More than 80% of important metabolic reactions are very well predicted. This methods, this prediction is based on similarity indexes. We should not forget also the formulation design. So we should not forget the importance of excipient, of toxicity of excipient. Excipient within principle should be pharmacologically inactive, may cause toxic effect in children, for example, due to different metabolism and pharmacokinetics with respect to adults. And steps stand for safety and toxicity of excipient for pediatrics. This database and tools were developed in collaboration by European and United States Pediatric Formulation Initiative. The STEP database aims at improving data collection on excipient toxicity and tolerance in children, and it allows searching for excipient and by excipient. The excipient properties, along with the toxicity data in children, depends upon the metabolic reactions. And some examples are reported in these slides. For example, aspartame, that is a sweetener used in tablets and liquid sugar-free formulation. And as you can see in this slide, the adverse effects are related to the release for cleavage of phenylalanine, which can be harmful in pediatric patients affected by phenylketonuria and rare hypersensitive reaction cross-reactivity with sulfonamide. Another excipients which should be taken in consideration is benzyl alcohol, which is used as preservative in several medicinal products and solution, and due to accumulation of metabolism in blood and brain toxicity, include metabolic acidosis, neurotoxicity, and gasping syndrome. We are trying to include some data in developmental toxicology and in prediction toxicology. One is the developmental toxicity model, Kaiser, last version, which is based on binary classifier single models based on more than 600 compounds. And developmental reproductive toxicity library, PGE, which is based on binary classifier single models with more than 2,000 compounds. And finally, developmental toxicity consensus model by toxicity estimation software tools, which is a consensus model from 4 other individual predictions. So just to give an account of the results related to metabolism related toxicity, benzyl alcohol, for example, which is a preservative compound used in formulation, shows toxicity in terms of metabolic acidosis, respiratory and central nervous system symptoms, associated with the use of this alcohol in intravenous fluids and heparin flash solution, and for this is recommended exclusion for newborn. The prediction of database is that benzyl alcohol can undergo 2 processes of oxidation catalyzed by an enzyme, by alcohol dehydrogenases, for providing for yielding benzoic acid, which is then conjugated by glycine to give epuric acid, which is a form of extraction. Unfortunately, conjugation with glycine in adult substantially decrease in newborns, and prediction said that you should not use this in newborns due to this metabolism-related toxicity. With aspartame, which is a sweetener, 3 major metabolic pathways in intestinal, lumen, and mucosal cells give the release of aspartate, which is a cytotoxin, methanol reaction, releasing methanol, which is a neurotoxin, and phenylalanine in portal blood. As a source of phenylalanine in blood should be totally avoided in children affected by phenylketonuria. So ontogenesis of metabolism and metabolism-related toxicity could be well predicted before starting with new formulation, for example, new drug formulation for children. So just some perspective that databases related reliable QSAR model and in silico tools may contribute to advance in discovery and optimization of medicines for children, and reliable property-based computational approach to the understanding of age-related drug metabolism and toxicity may contribute to progress in pediatric drug formulation design. Just I would like to thank my colleague, especially Professor Orazio Nicolotti and his co-worker, Rosa Purgatorio, Marco Capto, and Nunzio Denora from my department at the University, Department of Pharmacy at the University of Study Aldo Moro in Bari, and Professor Margheriti and Giordano for the Department of Biochemistry for Human Oncology, and all of you for your kind attention. Okay, this is what happened when we have, let's say, online presentation. Professor Altomare was unable to connect, so we had to use this kind of presentation, and this was the best that we learned to do up to now. We will get, I think everyone will get much more proficient in using these tools as these things goes on. So we are almost on time. So let's go to Professor Marushko. I hope that I said it well. And you are welcome to present.
Okay, thank you very much for your introduction and for invitation to participate in EPTRI Open Meeting. In my presentation, I would like to light up the information about our organization as pediatric institution. And secondly, placenta research implemented by our institution for a long time in relation to children's health, which can be applied in the future in different projects, including EPTRI. Ukraine is active, next please. Ukraine is actively involved in the EPTRI project under scientific guidance of the State Institute of Pediatrics, Obstetrics and Gynecology of National Academy of Medical Sciences of Ukraine. Next, please. Collaboration with EPTRI began in March 2019 after signing the agreement by director of the institute, Yuri Antipkin, and coordinator of EPTRI, Donato Bonifazi. Next, please. Since then, the Ministry of Health of Ukraine and Ministry of Education and Science of Ukraine granted the official support to EPTRI by signing the political support letters. Next, please. On the way to integrate national scientific community in pediatric research and through promotion in the country, 6 main Ukrainian research organizations had expressed their interest to participate in EPTRI project, and under chair of the institute organized themselves into the Ukrainian Joint Research Unit named EPTRI UKR. Next, please. Institute has long-lasting history of its activities, was founded in 1929, and at the moment is a leading and unique institution in the country in the field of maternity and childhood. It provides basic and applied research in the field of maternal and children's health in the country. Next, please. The main scientific activities in the field of pediatrics are development and implementation of new technologies to support children's health, research of broad range of different children's diseases, including autoimmune allergic disorders and orphan diseases, prenatal protection of the fetus and newborn, including prenatal diagnosis of congenital malformations and their early surgical correction, impact of negative environmental factors in the health of children. Next, please. Institute is operating as the largest referral pediatric hospital in Ukraine, relevant to pediatric research and an active center of clinical trials, has modern laboratory services and 6 specialized medical centers. Center of Allergology with diagnostics of drug allergy, Center of Children Hepatology, Rheumatology, Gastroenterology, Center of Orphan Diseases and Family Planning. Next, please. It also has obstetric and gynecological clinic providing high quality medical care for women of fertile age with increased risk of pregnancy and reproductive health. As a perinatal center of 4th level, institute provides high quality obstetric and neonatal care in Ukraine. Next, please. Among different scientific projects, institute performs research of placenta. Next, please. One of the long-lasting research conducted by the Institute is a study of the impact of Chernobyl accident on placental tissue with monitoring of structural changes depending on the accumulation of radionuclides. Were conducted morphological, immunohistochemical, and electron microscopic studies of features of the fetoplacental complex that exposed to low dose radiation by incorporating radionuclides into placenta. Concentration of radionuclides range from 0.5 to 4.8 becquerels per kilo. The research is continuing at present. Next, please. The obtained data were recorded to the computer program certificate of placenta. In total, more than 400 placenta had recorded to the program. The certificate of placenta includes passport data, histological and compulsory radiometric examinations. Certificate of placenta was legalized and included by the Ministry of Health of Ukraine in the list of laws related to the accident at the Chernobyl nuclear power plant. The main aim was to use collected data for further monitoring of children's health and study of risk factors of growth and development of children population after Chernobyl accident in Ukraine. Next, please. Study revealed morphological changes of proliferative nature that were correlated with a high level of expressed of carcinoembryonic and proliferative cell nuclear antigens, as well as pro-apoptotic factors, specifically in placenta with incorporation of radionuclides of 4.8 becquerels per kilo and more. Apoptotic index was 3 times higher compared to the control group. Proliferative changes were observed in syncytial trophoblast, stromal elements, mid-calf and terminal villus of endothelium. Next, please. In general, studies showed negative impact of low-dose radiation during 30 years after Chernobyl accident on health of children population and reproductive health of women, specifically those permanently resides in radiation-contaminated territories, noted an increase in malignancies in children and deterioration in reproductive health of women. Next, please. Since 1986, the Institute has been working with support and in collaboration with different countries, from which the greatest participation of Japan, Japanese scientists collaborated even since the beginning, visited radiation contaminated regions affected after Chernobyl disaster, help us with new technologies and diagnostics and treatment of affected children and pregnant women. Among many publications and presentations should be highlighted morphology and immunohistochemistry of placenta after 30 years of Chernobyl accident, which was presented on 4th Shiman International Symposium in 2018 in Japan. Next, please. Institute continues to research underlying placenta biology, particularly in exploring the use of placental biomarkers for the prediction of the onset of pregnancy and neonatal health issues before they manifest, restriction of growth and development of pathology in early life of children. Institute conducts research in the direction of placenta-derived stem cells with the determination of the effects of chronic hypoxia and internal irradiation on the structure of stem cells, pursue the architectonic of stem cells in the tissues of the placenta barrier, and regenerative properties of placenta cells. Resuming my presentation, I would like to emphasize that collaboration with EPTRI project will contribute to development of pediatric science and improvement of life quality of sick children who need special care in European community and in Ukraine. Next, please. Thank you for your attention. Hans.
Thank you, Professor Marusco. Just because we are very short in time now, we will go straight to the next presenter, Professor Mezzolani. We will try to have some question at the end of the session if we are still in time. Professor Mezzolani, please take you the microphone.
Hi, I'm doctor, not professor. Hi everybody, I'm Alessandra Mezzolani from the National Research Council of Italy, Institute for Biomedical Technologies Segrate, Milan. And I present the study Autism Spectrum Disorder, Linked Read Sequencing Reveals New and Undetected Variants. Next, please. No, sorry, okay. Autism is a complex neurodevelopmental condition that manifests within the first 3 years of age and lasts for the lifetime. It's characterized essentially by limited social interactions, communication impairments, and restricted interest and repetitive behaviors. From the epidemiologic point of view, the prevalence of ASD is about 1 to 68 with a strong male prevalence. Indeed, the male-female ratio is 5 to 1. As for etiology, it is complex and not completely understood. Indeed, although genetics play a key role in autism, causative or associated genetics variants have been detected only in 30% of patients, and thousands of genes are involved. For this region, a gene-environment interaction has been proposed for its pathogenesis. Since most of the genomics studies have been performed by GWAS or exome sequencing, here we test the hypothesis that variations in non-coding and or inaccessible regions could be involved in autism. And for the first time, linked read whole genome sequencing of patients with autism is used to assess discover and phasing disease-associated regions unmappable by short-reads next generation sequencing. These new variants, if validated on large scale of patients with autism and or on public autism genomic database, can be integrated in interactome studies to understand the pathogenic mechanisms and could represent new biomarkers useful to design diagnostic protocols and kits and for patient stratification and personalized medicine. For materials and methods, 10 patients were considered, 10 males and 3 females, including 2 couples of male siblings and 1 couple of male-female siblings. And how molecular weight DNA was submitted to 10x Genomics microfluidics partitioning and barcoding technology by the Chromium controlled platform coupled with the Chromium Genome Library Kit, and then to Illumina library preparation and whole genome sequencing. 10x long ranger pipelines and loop genome browser were used to find and phase single nucleotide variants, such as insertion deletions and single variants across more than 9 megabase haplotype blocks. In comparison to 1,000 genomes, genome aggregation database, and NHLBI ESP populations. Genes affected by variants were compared with those already associated with autism. That we found in Simon's Foundation Autism Research.
Hola.
Hola.
¿Qué tal? ¿Estás estudiando, guapa?
No, porque yo a mí se consigue.
Something is. Hello?
Jordi, Professor Manera, please. Good, it's closed. Go ahead.
So genes affected by variants were compared with those already associated with autism. We found in the Simon Foundation Autism Research Initiative, SFERI database, large-scale sequencing studies, and bioinformatics predictions. In this slide, we show the quality control of linked read analysis. And for instance, we report the sample 1. The input was 1.5 nanograms of high molecular weight DNA. And the SNP phase were 99.1%, and the longer phase blocks was about 6 million, more than 6 million nucleotides. The large structural variants calls were 18, and the short deletion calls more than, so almost 4,000. As for results, on the left, we listed the number of variants classified in total genes, total variants, annotation within genes, annotation out of genes, existing variants, and new variants. While on the right, we listed the type of variants, such as UTR variants, in-frame insertion, protein altering variants, missense mutation, and so on. As for variant consequences, on the left, we reported the type of variants we found. About 50% were intron variants, about 30% intergene invariants, and about 10% non-coding transcript variants. And among the coding variations, 58% were missense variants and about 35% synonymous variants. Sorry, next, please. Hello, the next. Okay. Here we reported the genes containing SFARI annotated and non-annotated SNPs distributed into SFARI categories. And on the left, we show genes containing SFARI annotated SNPs distributed into SFARI categories, where S is syndromic variation, and 1 is the class of highly associated variants. Class 2 is medium associated variant, and so on. On the right, we reported genes containing SFARI not annotated SNPs. So we showed how many genes already annotated in SFARI, but the variation is new. In conclusion, for the first time, we applied the linked read sequencing technology for studying more deeply autism genomics and discover new autism-associated variants. This powerful approach allowed to decipher the genomics heterogeneity in autism and highlighting variations otherwise undetectable by classic next-generation sequencing. Thanks to the study design, we also identify homozygous variants within genes or regulatory regions common to 3 couples of affected siblings. Further studies will be performed to validate the new autism variants in a large size of autism samples we already have in our lab and we can find in public data. The new biomarkers will be used to stratify patient population, and moreover, data integration will be also performed to identify pathways and gene networks involved in the disorder to understand disease mechanisms and design target-driven personalized treatment. A further sophisticated bioinformatics analysis of the results can discover the interactomes in which these variations are involved, leading to understand the autism pathogenic mechanisms and predict new associated genes. Possible development and application of dedicated artificial intelligence performed at CNR ITB can accelerate this analysis and integrate our data with the public genomic data of ASD. This approach can be applied to many other pediatric diseases with unknown genetic causes. I thank the co-authors, Francesca Anna Pupaioli, Ettore Mosca, Noemi Di Nanni, Paride Pelucchi, Luciano Milanesi, Maria Elisabetta Raggi, and Laura Villa. The first are from National Research Council Institute of Biomedical Technologies, and the last 2 from the Scientific Institute Eugenio Medea, Bosisio Parini, Italy. The projects we thank are the European project GEMMA, EPTRI, and CNR Biomics.
Thank you very much, Dottoressa Mezzalani. We have really short time, so we will go straight to the next speaker, that is Professor Burkhardt. Are you there? Professor Burkhardt? Hello? Professor Burkhardt, we don't hear you.
Hello? Can you hear me now?
Okay. Yes, now it's perfect. Go ahead.
Great. Good morning, everybody. Many thanks for the opportunity to present our recent research at this meeting. And in my today's talk, I'm going to present a child-appropriate multiplex mass spectrometric assay solution, which allows for the reliable monitoring of two major regulation systems, the renin-angiotensin-aldosterone system and the adjoining kinin-kallikrein system, and both by utilizing low sample volumes. The next slide, please. On this slide, you see the common presentation of both systems. On the right-hand side, the renin-angiotensin-aldosterone system. And here the angiotensinogen is cleaved by renin to angiotensin 1. And this decapeptide is converted into angiotensin 2 by the angiotensin converting enzyme, which is, amongst others, a prominent target in pharmacotherapy of hypertension, for example. Angiotensin 2 itself interacts with the angiotensin receptor and also affects aldosterone. The second system, less investigated than the renin-angiotensin-aldosterone system, is shown on the left. In the adjoining kinin-kallikrein system, kininogen is converted to bradykinin, which acts on the bradykinin 2 receptor, or is cleaved into its inactive metabolites also by the angiotensin converting enzyme. And these endogenous peptides and humoral parameters within the renin-angiotensin-aldosterone system and the kinin-kallikrein system play a key role in acute attacks of angioedema, arterial thrombosis, other cardiovascular diseases like hypertension or heart failure, but also in renal disease and adrenal insufficiency, for example. And additionally, their involvement in endocrinological diseases like diabetes or neurodegenerative diseases like Parkinson have been described. Further, this system have been implemented into inflammatory diseases like liver, pancreas, or bowel disease. Here, for example, morbus Crohn or colitis ulcerosa, but also in respiratory diseases, epilepsy, and depression. Although very little is known by now concerning these systems in children, it is known that both systems additionally undergo substantial developmental changes during childhood. Next slide, please. This busy slide shows that especially research during the last years in adults found the system to be much more elucidated and complex as it is illustrated nowadays in many textbooks. On the right, we see again the renin-angiotensin-aldosterone system. And here the rate limiting step of the system involving renin and subsequently also prorenin is highlighted in darker blue. One of the effectors, aldosterone, as well as precursor and its main metabolite are shown in lighter blue in the center of the slide. While on the left, the kinin-kallikrein system is again depicted. The small blue boxes indicate the elements of both systems that are commonly reported, and I have shown this to you on the slide before, while all other boxes show peptides and humoral parameters being currently intensively investigated in adults. For example, the research results on angiotensin 3 in animals and adults indicate that this peptide might be at least as vasoconstrictive as angiotensin 2. And in our current situation, you might know that the coronavirus interacts with the angiotensin converting enzyme 2, which is also embedded into this system. The interplay of the cascade of this cascade has never been comprehensively investigated in children. And an often cited argument is that the required blood volume for such a comprehensive investigation cannot be collected in children ethically. However, an understanding of the interplay among the complex biochemical cascade might either allow relevant insight into the etiology and pathology of the diseases, or new insight into the mechanism of drug action. Next. So nowadays, the investigation of those complex biochemical parameters is often limited by the blood volume that is available. Next slide, please. As you all might know, the European Medicines Agency has published a paper on ethical consideration in pediatrics. And in this paper, also recommendations on the maximum blood volume that should be collected in pediatrics is given. On the upper right, I have arranged an overview what this means to a newborn or a neonate. For example, 3 milliliters of blood at a single investigation for a newborn with a body weight of 4 kilograms and 9 milliliters within 4 weeks. And since also in recent pediatric trials, often the same analytical assays as the ones applied in adult studies were used, it gets obvious why comprehensive pediatric studies often only sample one pharmacodynamic parameter. And this most likely also only once during the whole study period. Please click. Next. On the bottom, I have selected 2 ongoing studies in children as an example for you. And what we get to know from the official text on clinicaltrials.gov is that these parameters are sampled once due to the restricted blood volume. But how shall we get better knowledge on the renin-angiotensin system if often only one parameter is monitored in the younger age groups? And this is done frequently in a single investigation only. So please click. Therefore, innovative and reliable bioanalytical tools are highly required, which facilitate the comprehensive investigation of those endogenous peptides in small human sample volumes. Next slide. Consequently, we have put our effort into the development of a tailored multiplex bioanalytical platform to overcome the aforementioned limitations. The established platform bases on a high-performance liquid chromatography coupled with mass spectrometric detection, and this technology is regarded superior as known limitations by immunoassays, for instance, can be circumvented. A main focus, beside the highest possible coverage of humoral parameters into the platform, was the setup of the platform with small sample volume to enable a comprehensive investigations also by repeated sampling in all pediatric age groups. Its broad calibration range ensures its application to healthy and diseased population, as most humoral parameters are known to alter into the opposite direction if a healthy or a diseased population is investigated. And finally, the platform was established on a high throughput approach to enroll it into clinical studies. And the latter is also promoted by the fact that the platform is fully validated according to international agency requirements and embedded into a good clinical laboratory practice compliant environment. Next. So this platform supports a maximum gain from the humoral blood samples or the blood volume that is available. But what can we exactly gain by this ready-to-use platform? The next slide, please. This multiplex bioanalytic platform will monitor not only one biomarker or surrogate parameter in a small sample volume, like, for example, NT-proBNP in cardiovascular diseases. The system enables a deeper and comprehensive insight into the main cascade and monitor it coherently. In its current extent, it allows for the comprehensive investigation of 7 parameters in the renin-kallikrein system. This is on the left again, and 14 parameters within the renin-angiotensin-aldosterone system on the right. And by applying this platform, we will be able to monitor precursors, main metabolites, as well as their main substances, as well as their metabolites in low sample volume. Next slide, please. Let me once again use this slide from the beginning of my presentation to illustrate what we can analyze by the established low volume bioanalytical platform. Please click. All wet marked peptides and humoral parameters are enclosed in the determination. Next. And due to the low total sample volume of 500 microliters, 21 parameters acting in both systems can be analyzed, even by repeated determination in, for example, neonates. This platform facilitates the comprehensive investigation in healthy and diseased children without infringing ethical considerations. And moreover, the low sample volume makes it possible to collect blood in children more than once, and therefore empowers researchers to evaluate the meta-rating renin-angiotensin-aldosterone system and its adjoining renin-kallikrein system. My last slide, please. I have shown you the developed and validated multiplex bioanalytical platform, which bases on highly reliable mass spectrometric bioanalysis. And it allows for the comprehensive monitoring of two major and important regulation systems, and subsequently enables a deeper and coherent insight into etiology and pathology of, for example, cardiovascular, renal, endocrinological, inflammatory, and respiratory diseases. It also supports the investigation in the context of the COVID-19 disease, of course, and due to the low sample volume, the system can be applied to all age groups, children, adults, and elderly, without infringing ethical considerations. And therefore, this platform might help to identify new promising drug targets, but for sure facilitates better understanding of important diseases. So finally, we hope to further enroll this platform very soon into further and future research. Next. And therefore, many thanks for your kind attention, and also many thanks to my colleagues and co-workers at the Institute of Clinical Pharmacy and Pharmacotherapy in Düsseldorf.
Thank you, Professor Burkhardt. In the meantime, if anyone wants to put question, you can put down in the chat for the panelists so they can see them. I have two questions for Dottoressa Mezzalani. One, if there is any association of the autism with HLA genes in your panel.
At the moment, we have analyzed just the groups of genes, and we classified them in two categories of by functionality, by function. The work is in progress. We are now analyzing the pathways and the groups of the group of genes. At the moment, we have analyzed just the variation, the type of variations.
Okay, so the other question was which class of non-coding RNAs were altered in the genome sequencing for the ASD patients?
The non-coding RNA.
Yeah.
We don't know at the moment. We have no information about this one, this kind of question.
Okay. Well, with a lot of things to do. I have now another question for Professor Burchard. Do you think that it will be feasible to centralize the samples related to this topic in your center as a reference center?
Of course, we can act as a central laboratory for Europe, and can be also act in a collaboration to ensure that not known assay variations interact with the results, or so yes, of course, we can act as a central laboratory, and of course, we also can ensure the analysis on a broad base.
That's a plan then. See, EPTRI can be of help there. Thank you very much. I don't have other question. So let's go to the next speaker, that is Beate. Oh, my French is very bad. Aurich.
Hello, thank you, Giovanni. So thank you for the invitation. And today I will be providing you with an introduction to the topic of drug safety in translational pediatric research, and the question on how to anticipate and manage treatment-related risks in clinical trials. So the next slide, please. And the next, please. So the question really is, how is knowledge on treatment-related risks included into a pediatric translational research program or a trial? One may wonder. Next slide, please. So to provide you with an introduction to this topic, which is obviously quite vast, I will be touching on anticipating treatment-related risks in children using pediatric safety profiling, pediatric protocol and risk management, the safety data review cycle, and drug safety in pediatric translational research. Next slide, please. So in pediatric research, treatment-related risks can be, to a degree, anticipated by using safety profiling. Safety profiling builds on two blocks of information mainly. One is the safety specification of the study drug, shown in the first block on the left. And the second is the data on the pediatric study population, shown in the gray box on the right. The safety profile of a study drug includes the safety data from all age groups and all indications, including, for example, non-clinical and clinical data. And the pediatric population-specific data includes, for example, things like age group-specific reference ranges, comorbidities, co-medications, and as mentioned by the previous speaker, the limitations of invasive procedures, such as blood sampling. The data on the general and pediatric safety profile of the study drug and the specificities of the pediatric age group then provide the evidence of the pediatric safety specification shown in the box on the bottom of the slide. This box summarizes the identified and the potential treatment-related risks, and importantly, the missing safety information in children. So to summarize, safety profiles are population-specific, as you can see, and they help with anticipating treatment-related risks, identifying missing safety data, and planning resources and priorities in pediatric research. So this may apply to a whole research program as well as a specific study protocol. Next slide, please. So in the context of the development of a pediatric protocol, pediatric risk management and pharmacovigilance do require an understanding of this pediatric safety specification as shown in the blue box on the left of the slide and as illustrated in the previous slide you've just seen. In addition, the characteristics of adverse drug reactions in the pediatric study population and population-specific reference ranges are required as described in the orange box on the right of the slide. In the center, in the green box, you can see that several protocol sections are informed by the pediatric safety specification, including those related to risk management, such as the exclusion criteria, for example, or the safety data collection for safety monitoring, and those pertaining to pediatric pharmacovigilance. Pediatric pharmacovigilance as such collects, analyzes, and follows up on general safety data, for example, adverse events, suspected ADRs, abnormal laboratory values, or vital signs. In addition, targeted safety data collection based on the pediatric safety specification will contribute in the context of a pediatric study to characterize identified and potential treatment-related risks. So therefore filling some of the knowledge gaps we have on drug safety in children, and this is really important. So in summary, pediatric protocol development requires a knowledge on pediatric risk management and pharmacovigilance. Next slide. Now I just want to take a little step back to touch on some of the fundamental concepts of drug safety. Drug safety is a cross-functional activity requiring the input from many different specialists, as we've seen this morning. And these days, safety physicians who work in the area usually start to get involved just prior to the first in-man studies, and they continue to monitor drug safety profiles throughout the life cycle of a drug, so way after it has been on the market. And as you can see on the slide, safety data review is a continuous process. It includes pharmacovigilance and risk management. Pharmacovigilance as such has been defined by the WHO as the detection, the assessment, understanding, and prevention of adverse drug reactions or any other treatment-related problems, such as, for example, medication errors. And in the following, once a risk has been identified, we use risk management to ensure that the benefits of treatment still outweigh the treatment-related risks as much as possible. Risk management and activities include, for example, the communication in the product label, which is the kind of traditional way of communicating risk and reducing them. And they get reassessed ever so often to see whether they are effective using pharmacovigilance techniques. And in addition, existing knowledge on adverse drug reactions is constantly re-evaluated once new safety data becomes available. And thus refines the understanding of, for example, risk factors. And this goes for all populations, including children. So in summary, safety data review is a continuous process throughout the life cycle of a product and includes both pharmacovigilance and risk management activities. Next slide. Thank you. So this is a bit of a busy slide. Sorry for that. The idea is to show how pediatric safety data from all phases of drug development, so from the preclinical stage to the observational post-marketing stage, is constantly contributing to updating information on the safety profile and the risks. So it's for example, if you look on the very left of the slide, you can see that protocol development for a first in children study will be informed by the existing knowledge on the risks of the drug. Data from that study will then feed back into the next study and so on. So it's a continuous back and forth of information. And in safety, we use the safety profile then informs the so-called risk management plan. You may have heard of this. So the risk management plan then feeds into the protocol using a consistent approach across, for example, multiple protocols and multiple activities to have coherent data collection and assessment. So in summary, data collection and analysis and translation of pediatric research is a continuous process, building on existing data and updating the safety profile and the risk management plan as needed. Next slide, please. Thank you. So in the context of EPTRI, as you've seen in the previous presentations, safety information on children is assessed and examined and is a topic. And I've listed on the slide just a few examples of the work packages where information on the safety profile may inform the studies then performed in the different work packages. And as we all know, the information, the learnings from the work packages then will feed into pediatric studies and subsequently into clinical practice. So in summary, pediatric safety profiling in translational research may inform non-clinical and clinical studies as well as clinical practice. Next slide, please. Okay, so just to summarize, the pediatric safety profile forms an integral part of translational research in children. It helps with defining and anticipating treatment-related risks, identifying knowledge gaps concerning treatment-related risks in children, and planning of resources and priorities in pediatric research. It will help with informing non-clinical and clinical studies as well as clinical practice. The pediatric drug safety assessment is a continuous process throughout the life cycle of a medicinal product or a device. And it includes pediatric pharmacovigilance and risk management in clinical studies and clinical practice, with the aim of informing children, parents, and healthcare professionals about the benefit-risk balance of treatment. Thank you for your attention.
Thank you very much. I don't see yet any question. We will wait just a moment to see if anyone wants to write something.
Yes,
it's a comment for all of the panelists. I see a lot of possibilities to translate this research into diagnostic or therapeutics. One of the things that the EPTRI infrastructure wants to do is to facilitate this passage from something that can be seen only as a laboratory research to something that can be used in the clinics.
Yes.
So one of the services that EPTRI will have, it is just supporting both as, let's say, expert advice and in a factual manner how to use this in a proficient and economically positive way. Okay.
Oh, this is,
but this is for all the panelists, right? You want that? Okay, good.
Oh yes, of course. Yeah. As to basic to translate the clinical is grateful. It's great.
Yeah, one of the question will be how big the sample of intestinal mucosa needs to be to allow you to do the testing with the flow cytometry, or is there another way?
No, it is enough also one small pieces of biopsy, because we work in a very sensitive way and with also low amount of cells. Normally, 2 biopsies are taken for diagnosis, and 1 or 2 comes in the lab for the research.
Okay. Well, let's try to have maybe something less invasive than a biopsy. That is always better.
Oh yes, there is. We have also another challenge in oral short-term oral challenge. We look also at peripheral blood, and there is another presentation that offer another tools to look at peripheral blood also.
Perfect. So you see, we are already working that way.
Okay.
The next 2 speakers, they have problems with the connection. One is not able to connect with the microphone, and the other one is not able to answer and enter in the webinar. So we will have to put the audio file again on the screen. Hopefully, you can work your volume if the spoken presentation is too low, but it should work. The other, let's say, administrative point is that the meeting has an opportunity to publish all the presentation as articles in a special issue of pharmaceutics that has a very good impact factor. It's an open access journal. And so we invite everyone to prepare and get possibly article related to the presentation ready for submission. Obviously, we'll go under peer review, so no one is sure to get the paper, but if we reach the target of at least 10 papers submitted and accepted, the journal will publish in a paper format, so you will have also the booklet to keep, which is always nice to keep in the library. So that's the information. And now I'm to ask Mariangela if she is ready with the next files, which is taking some time anyway.
Yes, we are ready. Okay, thank you.
So let's start with the presentation from Professor Evelyn Jack-Egrain. I'm sure I said it wrong, but she is one of the founder of EPTRI. Start if you can.
this presentation, in silico modeling and simulation for dosage selection in pediatric patients, including neonates. This work and this presentation were prepared by Stéphanie Leroux, Wei Zhao, and myself. I am professor in pediatric pharmacology in Hôpital Robert Debré in Paris. Population pharmacokinetics, modeling and simulation were conducted to evaluate the pharmacokinetics and variability of currently used drugs in pediatric patients of different age groups, from neonate to adolescent with different disease. In summary, we tested the following drugs: ciprofloxacin in neonates and patients with sickle cell disease, and the corresponding studies are presented in the following slides. Vancomycin in neonates, but also metronidazole in neonates, ceftazidime in patients with cystic fibrosis, and additional drugs, ganciclovir, fluconazole, and cyclosporine. Our aims were to recommend adapted drug dosage in children based on innovative methods. Complex modeling methods of population pharmacokinetics, pharmacokinetic pharmacodynamic, and physiologically based pharmacokinetic modeling are currently used, requiring analysis of covariates that may participate to variability in drug disposition and responses. As summarized below, drug concentration, characterizing disposition pharmacokinetics, are linked to biophase distribution, biosensor process, and biosignal flux with a transduction to response, linking drug administration and concentration to drug response. Our objectives were to develop pharmacokinetic modeling and simulation methods to investigate the impact of key potential covariates on drug disposition of currently used drug in pediatric patients. The principle of population pharmacokinetic modeling is the following. First, concentration data are used to describe the pharmacokinetic parameters and variability. Evaluation of key pharmacokinetic variables is followed by identification of relevant predictors and simulation of clinical scenarios. We focused the analysis of variability on age and age group, from neonates to children, with different pediatric diseases, neonatal infection, sickle cell disease, cystic fibrosis, hemato-oncology disease, and we started working on the potential impact of ethnicity. I am going to show you the results that we obtained when studying ciprofloxacin in sickle cell disease. We studied 60 patients, 4 months to 18 years, with the disease or with other various diseases. We determined the ciprofloxacin clearance that was dependent on weight, creatinine concentration, but was 52% higher in patients with sickle cell disease compared to non-sickle cell disease patients. And we performed Monte Carlo simulation, as shown on the right panel, showing that the dose required to target the AUC over MIC of 125 required for antibacterial efficacy was, as expected, higher in patients with sickle cell disease. Modification of dosing regimens is therefore recommended. We also investigated the pharmacokinetics of ciprofloxacin in neonates. Very limited data on 10 patients 3 months to 1 year was at that time available in the literature. So we investigated the pharmacokinetics in 60 neonate and infants treated with 10 milligram per kilogram either BID or TID. And as you can see, ciprofloxacin concentration were highly variable when presented according to time since last dose. We developed a population pharmacokinetic model where clearance was dependent on current weight, on a function of age, and a function of renal function. As a result, we've shown that 90% of neonates with a post-menstrual age lower than 34 weeks and receiving 7.5 milligram per kilogram BID, and 84% of neonates with a post-menstrual age higher or equal to 34 weeks and receiving this time 12.5 milligram per kilogram TID would reach the IUC over MIC target of 125. We also demonstrated that an appropriately constructed population pharmacokinetic model using opportunistic sample can reliably estimate important pharmacokinetic parameters needed to support individualization of therapy in neonates. And this is shown in the on the right, on the left side, with the results of the full model, the model based on only opportunistic sample, and the model based on trial samples. Our last example is related to pharmacokinetics of vancomycin. We perform a very large meta-analysis of 1,631 neonates and infants that were highly variable in terms of weight, gestational age, post-menstrual age, postnatal age, and birth weight. And we analyzed more than 4,000 vancomycin concentration. With this data, we developed a population pharmacokinetic model. We modified dosage recommendation. The target vancomycin AUC is recommended to be 400 milligram hour per liter. And you can see on the screen that the reference dose of 15 milligram once daily is given in patient less than 29 weeks post-menstrual age, and that this concentration on the table is not, the dose is not high enough to reach the target. And therefore we modified the dose and simulated the AUC that would be obtained by increasing the dose in the lower group of patients and adding in the 3 group of neonates with different postmenstrual age, a loading dose of 25 milligram. And in such a case, the AUC is higher in the lower group, reaching 400 milligrams hour per liter. As a conclusion, population PK and PBPK modeling are of particular interest to explore drug disposition in children when data are limited and or the impact of the disease is usually unknown. Information on drug administration and concentration are needed, but in addition, in pediatrics, key covariates may include age, gestational age, postnatal age, postmenstrual age in neonates, weight, birth weight or current weight in neonates, and body mass index, different biomarkers of renal and hepatic functions, including also pharmacogenetic biomarkers, characteristic of the disease, hemato-oncology, cystic fibrosis, or sickle cell disease, and care and treatments that may include the impact of co-administered drugs, ventilation, and hydration. Thank you for your attention.
Okay. We are very sorry that Evelyn could not talk at this meeting, but we will have occasion to hear her live again. So next talk will be from Professor Gorovenko, and I think Mariangela is putting on. Perfect. Let's start.
Me and my team to introduce our research work, the correction of the drug dosage according to the changes in the weight of the young children during long-term treatment that can reduce the toxicity and the side effects of treatment. I'm pleased to introduce the whole team, Dr. Natalia Gorovenko, professor of Shupik National Medical Academy for postgraduate education, who led this project, Dr. Lilia Todoriko from Bukovinian State Medical University, Chernivtsi, Ukraine, who helped to collect the material, and me, Dr. Tatiana Sprinsian from Communicable Diseases Intensive Care Association in Cure, who will be the presenter, author of this research work. As all we know, that in young children, the accuracy of dosing is extremely important due to the requirement to keep the balance between the optimal efficacy and the risk of toxicity. Due to this, medicine are being prescribed per kilo of body weight. But some conditions, such as oncology disease, tuberculosis treatment, or the treatment of latent TB, some chronic diseases, require long-term therapy. And during this treatment course, young children can increase in the body weight extremely, especially the young babies under 12 months or children under 2 or 3 years old. But the question is, if we do the dose correction during the treatment course of such kids, or we just see the patient prescribe the approximate dosage and then leave the parents or caregivers to treat the child by themselves. You know that tuberculosis, active and latent disease require at last 6 months of therapy. Oncology can last for years. How often doctor adjusts the scheme of the therapy to the increasing of the body weight of small patient? Every month, every week, every year. So aim was to see if the adjustment of the prescribed dosage in very small kids under 2 years old during the treatment course, on the example of the treatment of latent TB infection during 6 months, can be beneficial in such patients to decrease side effects, to increase tolerability, and to increase adherence to treatment. We performed the comparative study in two groups of children with latent TB infection that required isoniazid treatment during 6 months. In the first group, we included 114 children who received their isoniazid in the liquid pediatric formulation in the syrup form with the recommended by WHO dosage 10 milligram per kilo once a day after breakfast. And the children from the control group received the same drug in the form of tablets. And due to the limited possibility to divide the tablets very accurately, we used the recommended by WHO table how to divide the tablet according to the range of the weight, not to the accurate body weight. As you can see, with the liquid formulation, it was very easy to give the exact amount of the required milligrams to any child of the, any body weight. And when we used tablets, very approximately the dividing could be used. For example, children under 5 received half of the standard tablet. Children with body weight range from 5.1 to 9.9 received 1 tablet, but it's obvious that they need in the amount of the drug was different. So we have found that 65 children from the main group, 57%, 61 from the control had increased in the body weight during the course of treatment during 6 months in more than 1 kilo, which required the dose correction. But the main increasing of the body weight during these 6 months was about 3 kilos in both groups, which showed that the dose correction should have been performed according to the standards of the treatment by WHO quite significantly. Actually, the only one formulation in liquid form, in syrup form, allowed the dose adjustment very accurately according to the body weight. That's why the patient from the both group were seeing the doctor every month and measured the body weight, but only the patients from the first group could receive the dose adjustments due to the increasing of the body weights. Also, all the parents or caregivers from the both groups were asked to fill the questionnaire to specify the scale from 1 to 5, the ease of treatment and willingness to continue the entire course 6 months. It's well known that adherence in treatment of latent TB infection is very low. Only 40% of the caregivers fulfill the whole 6 months course, which is very low level. So from our point of view, it was very important if the drug formulation could influence on adherence and be beneficial for the parent. On this slide, you can see that we have received significantly higher results in the group of the main study, where little patients had those adjustments. They had much better levels of bilirubin, of LT, of AST, also Timol test, which showed that they had less toxic effects comparing to the isoniazid in tablet form without dose correction. The changes in the blood chemistry were much more prominent in the children who received isoniazid in tablets and were increasing up to the completion of the course. Also, the tolerability of the treatment was defined as good in almost all the patients in the main group and only in 83% of the control. And significantly much more satisfactory level was shown in the main group compared to the control. Those findings allow to tell that the dose adjustments to young children with rapid weight gain during the continuous course of the chemotherapy allows decreasing side effects and toxicity, increasing the tolerability of the medicines, also increase adherence, which is essential in potential toxic agents. Liquid drug formulations like syrup or suspensions allow more accurate dose adjustment and should be considered as preferable child-friendly dosage forms for babies and infants over tablets and over dispersible tablets. We invite our colleagues to continue further research works in this area. Thank you very much for your attention.
Okay, so we can come back to the oral presentation with Professor Nunzio De Nora. Nunzio, can you hear us? Perfect.
Yes, I can hear you, Giovanni.
So
Can you
here
hear me?
your slide. Yes, perfect.
Okay. So thank you, Giovanni, for your presentation, and hello everyone. Before I start, I would like to thank the scientific and the organizing committee to give me the chance today to share with all of you this research study concerning the investigation and development of propanolol hydrochloride oral solid formulation designed for pediatric patients. Please, let's move to the second slide. Propanolol hydrochloride is a non-selective beta-adrenergic receptor blocking agent, and this API has a well-documented history of safety in pediatric therapy for the treatment of different cardiac diseases and also hemangioma, which is a vascular tumor of a childhood affecting about 1 in 10 infants. Currently, propanolol tablets are available on the European market in a dosage not suitable for children. But we have also oral liquid formulation, which are proposed as an alternative to solid dosage forms, having some benefits. However, this preparation is inconvenient to transport and store for more than 60 days after opening. In addition, this API has a bitter salty taste that does not facilitate high rates of compliance among children, especially in liquid formulations. Therefore, the development of a new oral multiple unit dosage form designed for pediatric patients would have the potential to deliver several advantages, including flexible dosing, improved patient compliance by masking an unpleasant taste, enhanced swallowability, and finally also a longer stability. Next slide, please. So the aim of this study was to realize new pediatric formulation of the bitter API propanolol with the polymer masking agent Eudragit APO, which has been supplied by Evonik company, using also an innovative prilling technology in order to improve the palatability of a dosage form and to be comparable with other commercial formulation given to pediatric patients. The obtained microbeads were characterized in terms of drug loading, efficiency of encapsulation, size, and also morphology. The in vitro drug release and also the electronic tongue tests of this formulation were also performed in order to evaluate the efficacy of this microparticular system in avoiding the bitterness in the mouth after administration and the complete release of the API in the stomach. Next slide, please. So let's move now to the methods, the experimental section of this study. As you can see in the table, 9 different samples were prepared by prilling concealing technique using the one encapsulator equipped with a single nozzle and processing the feeds with different ratio drug polymeric matrix in aqueous media. The microbeads were all characterized in terms of percentage of encapsulation efficiency, drug loading, and stability by using HPLC analysis, while size, morphology, and inner structure were analyzed by SEM analysis. FTER and DSC analysis were performed to deposit the compatibility and stability of the drug with the polymer. Release studies of this system were also performed in order to evaluate the release and dissolution of the drug in artificial saliva and also in simulated gastric media. Last was the electronic tongue measurements, which were performed to estimate the ability of this formulation to mask the bitter drug taste. So let's move to the other slide. And we can move to the results. And as you can see in this slide, 9 different samples were prepared using the prilling concealing technique. Most of them are characterized by high yield, except for the formulation F2N. We used the 2 nozzle and the diameter obtained of the microbeads measured by digital caliper or by optical microscopy were always homogeneous, ranging from 300 to 700 micrometers, and these size are appropriate to be swallowed by children. Most of the examined microbeads were characterized also by very high percentage of encapsulation efficiency and drug loading, effective for the administration of low and also high doses of propranolol. Next slide, please. As shown from SEM analysis, all the microbeads formulated are quite spherical and homogeneous. And this characteristic, together with the softness of prepared microbeads, could help to reduce the grittiness and mouthfeel, which are disadvantages that are often associated to multi-particulate. Moreover, the SEM analysis revealed a matrix with a radial or spongy structure with numerous pores that generated soft floating microbeads in aqueous solution. Let's move to the next one. DSC thermograms and the air spectra of the propranolol loaded in the microbeads revealed the presence of drug polymer interaction due to hydrogen bonds between the hydrogens of the hydroxyl and the second amine groups of the drug, which can be considered the donor, and the ion pair of the electrons of the oxygen on the carboxylate group on nitrogen of the tertiary amine of the polymer, which is considered to be the acceptor. So let's move to next slide, please. So release studies. These studies confirmed a prompt dissolution in simulated gastric media at pH 1.2 and a low release and dissolution of the drug in artificial saliva at pH 6.8. Mainly for the microbeads with a ratio drug polymer matrix 1 to 25, which is formulation F1N. As expected, the propranolol immediately dissolved in artificial saliva at pH 6.8, with about 89% of drug dissolved after 2.5 minutes, and the release rates of the drug and its dissolution from manufactured microbits are much lower than that of drug alone and statistically significant. Especially the sample F1N dissolved and released about 3% of the drug after 2.5 minutes at pH 6.8. That was about 27-fold lower than the percentage of propanolol alone dissolved at the same time and pH value. As a consequence, the unpleasant taste of the drug in this sample should be reduced. Next slide, please. So finally, here there are the results concerning the electronic tongue measurements, which revealed the ability of this formulation to mask the bitter drug taste, especially for the sample with the ratio 1:25 drug polymeric matrix, which is the formulation F1N. Considering this formulation, the total bitter taste scores are equal to 9% at 2.5 minutes of dissolution and to 20 after 15 minutes of dissolution, corresponding to a bitterness reduction percentage of 91% and 80% when compared to propanolol alone. These findings are in agreement with the assumption made in the release study of propanolol in artificial saliva, where the sample F1N was recognized as the best to cover the bitter taste. So in conclusion, we can move to the final slide. In conclusion, for the first time, matrix microbits loading propanolol were developed using an innovative pre-link concelling technique, and these microbits, these microparticles, have the potential to deliver several advantages, including flexible dosing, improved patient compliance by masking an unpleasant taste, enhanced swallowability, and finally very longer stability. So at the end of this presentation, let me thank all the colleagues of the pharmaceutical technology laboratories of the Department of Pharmacy, University of Bari. In particular, I would like to thank Professor Angela Lopedota and Antonio Lopalco, which contributed to this work. And finally, thank you for all of you for your attention.
Thank you very much, Nunzio. I don't see any question at the moment, so let's go on for the next speaker. And then if there are questions, we will put to you later on. So Professor Wiegand, I hope good pronunciation. It's yours, the microphone. Yes, go ahead.
Okay, awesome. Yeah, thank you. Hi, everyone. Before we get started, I'd like to thank the committee for the opportunity to present my subject. My name is Felix Wiegand, and I'm a PhD student at Fraunhofer Institute for Toxicology and Experimental Medicine in Hanover, Germany. My presentation today focuses on a technology for breast-triggered aerosol release and real-time determination of the delivered aerosol for preterm neonates. Next slide, please. When we treat someone by inhalation with an aerosolizer like the one on the right side, we obviously want to provide the patient effectively with as much aerosolized drug as possible. But as you can imagine, the aerosolized drug can only reach your lungs during inhalation. When you use a conventional system without a breast trigger technology, you have a waste of drug during exhalation. This means that the efficiency of the delivered aerosol to the patient depends on the inhalation-exhalation ratio. Now imagine, we treat a preterm neonate who inhales for 0.3 seconds and exhales for 1.2 seconds. Now we fill our aerosolizer with 2 milliliters of liquid medication. Of these 2 milliliters, only 0.4 milliliters are available to the preterm neonate, which is 20% of the original amount, representing a loss of 80%. Next slide, please. So we have an 80% loss of drug if we do not use breast trigger technology. However, if we use such technology, we can ideally reduce the loss to 0. Unfortunately, preterm neonates have challenging breathing patterns, such as a high breathing frequency of up to 60 breaths per minute, a low tidal volume of 4 to 6 milliliters per kilogram, and the short inspiration time of 2.2 to 0.4 seconds. Due to these challenges, there's no existing technology available which releases the aerosolized drug into the patient interface in a breath-triggered manner. On top of that, if we had such a technology, there's no standard test procedure to determine the aerosol output accessible. Next slide, please. What are we doing to address the problem? First, we are developing a novel modified nasal prong. This nasal prong has an integrated micronized valve. This valve allows direct and fast breath-triggered release of the aerosol at a targeted bolus, which increases the inhaled dose efficiency. Second, we are developing an appropriate test bench to measure aerosol output. The test bench is applicable for preterm neonate breathing patterns and also enables real-time measurement. Next slide, please. Our first objective is the nasal prong. On this slide, you see a schematic structure. The aerosol and breathing gas enters the nasal prong on the right side. The aerosol release is triggered by an integrated micronized valve, which is controlled by adding or removing a defined volume of gas. The preterm neonate is inhaling the delivered aerosol over the 2 nasal tubes, which are placed on the nose. The non-inhaled aerosol leaves the nasal prong on the left side. Next slide, please. Thank you. Our second objective is the test bench. This picture shows the schematic structure of the test bench with the test lung, the aerosolizer system, the ventilation system, the nasal prong with its integrated aerosol valve, and the 2 measurement zones for the inhaled aerosol and non-inhaled aerosol. The test lung simulates the preterm neonate breathing pattern. During a simulated inhalation, particles are sucked through the nasal prong into the measurement zone inhaled aerosol. Once there, these inhaled aerosol particles are first measured by the laser-based optical measurement unit using light extinction, followed by sampling the particles on the filter. Particles not sucked in are guided towards the measurement zone non-inhaled aerosol during a simulated exhalation. These non-inhaled particles are then measured by the second laser-based optical measurement unit, again using light extinction, followed by sampling the particles on a second filter. To determine the aerosol release efficiency, a ratio of inhaled and non-inhaled aerosol is used. This ratio is defined as the collected inhaled aerosol mass divided by the total of the collected inhaled aerosol mass and non-inhaled aerosol mass. The used parameters are described on the right side. The next slide, please. I will show you 2 videos to demonstrate the triggered aerosol release technology which we have developed. Please click to start the video. Thank you. In the left video, you can see the integrated aerosol valve in action. The response time of the valve to fully close or open is less than 20 milliseconds. In the right video, you can see the triggered aerosol release. The release duration and the release timing can be variably controlled via the valve. This means that if the aerosol is released early during inhalation, we can treat the lower airways, and if aerosol is released later, we can treat the upper airways. Next slide, please. Thank you. This diagram shows the aerosol release efficiency for non-triggered aerosol release and triggered aerosol release. Here you can see that the triggered aerosol release at 83% is more efficient than the non-triggered aerosol release at only 26%. This is equal to an increase in efficiency by a factor of 3.2. Combining gravimetric and optical detection using the test bench, we were also able to perform real-time measurements. Next slide, please. To summarize the aforementioned work, we have developed a modified nasal prong with an integrated micronized valve, which releases the aerosol as a targeted bolus, fast and directly into the patient interface. The breath-triggered release has an increased inhaled dose efficiency. We have also developed a test bench to measure aerosol output suitable for the breathing pattern of preterm neonates in real time. Thus, our breath-triggered drug release system enables administration of high concentration of aerosols despite the very challenging breathing habits of preterm neonates. Our next step is to further develop the modified nasal prong and the test bench approach. That's it for my presentation on a novel technology addressing breath-triggered drug release for preterm neonates. Next slide, please. Yeah, thank you for your attention. I'm now open for questions. Please also feel free to send me an email with any questions.
Thank you, Dr. Pullman. So there is any question? No, I see no further question. At least no one wrote anyone. Okay. I see
Okay,
that you use quite a lot of novel techniques. So the integration of this in the intensive care unit for neonates will be quite interesting. But also the translation of this on the market for the market of the medical device usually is not so easy to enter. Have you ever planned any kind of marketing for this device?
So yeah, this technology is in several projects involved, and then a European Union project and a German project, and yeah, we are trying to enter the market.
Okay, good luck, and we will be very happy to hear again from you. So
thank you.
We are. Go ahead.
Yeah, thanks. Thanks everyone, and I want to say stay safe out there.
Oh yes, we all need to stay safe. Well, we are at the end of the morning session. So finally, I can send you for lunch, but please be back for 2 when we start the section 6, EPTRI Outreach and Development. Thanks to all, and stay in touch and stay safe. Bye.